My talk, the outline of my talk is the following. So I will start by telling you a bit about small angle scattering. I'm not going to limit myself to neutrons. So I'm going to tell you a bit about X-rays and light also, and what you can do with it. And then I'm going to tell you a bit about SANS and ISIS. And then to finish, I'm going to give you some examples of SANS, and especially the use of contrast variation and contrast matching to characterize nanoparticles. So first, what is SAS and what can it do for you? So small angle scattering is a coherent, elastic scattering of radiation at small angles, whether the radiation is X-rays or neutron or light. It is used for structural characterization of materials. Conceptually and theoretically, SACS, SANS, and SALS are really very similar. The main difference basically stems from the physics of the interaction of the radiation and matter, while for X-rays and laser, it's an electromagnetic interaction, a type of interaction. For neutrons, it is a radiation between matter and a neutral particle. And basically, it is used to characterize different types of matter. So as I said, conceptually, it's really very simple. So all you do is basically you shine radiation on your sample, and it's detected at an angle 2 theta that is comprised between 0.3 and 5 degrees. This gives you a wave vector that is smaller than 0.5 inverse angstrom. Scattering is a diffraction, actually. It's just this type of diffraction where the objects are much larger than the wavelength, especially for X-rays and neutrons. And obviously, as a diffraction, it obeys Bragg's law, which is, as you can see, lambda equal 2d sinus theta. That basically gives you a q is, or the wave vector is equal to pi over d. So basically, in the simplest of terms, if you're looking at larger structures, you have basically to look at them at small q. What type of samples do we look at using SAS? It's literally anything you can think of. It's really widely applicable to look at solids, liquid, gases, different types of structures, whether it's amorphous or crystalline or anything in between, and pretty much any type, any kind of material. It is usually used complementarily or alternatively to microscopy. And as you can see on this graph here, so basically you can look at polymers, proteins, viruses, nanoparticles, micelles, nanocomposites, powders, yeah, pretty much anything. So from an instrumentation point of view, SAS instruments are conceptually quite simple. So basically all you need is the source, and that's usually, for X-ray, it's either an X-ray generator or a synchrotron. For neutrons, it's usually a spallation source or reactor. While X-rays can be done in the lab, you can have a bench SACS kit. Neutrons, for now, we don't have neutron, we don't have SANS bench kits. You need to go to a large scale facility. Obviously, you need to define the wavelength you want to look at your systems with, and usually you use either a monochromator or a chopper. So a monochromator, if you're using a monochromatic beam, that's usually the case in reactor sources. ISIS, we use choppers because we have wave beams and we use time of flight instruments. Obviously, you need to collimate your beam. And for that we use collimating optics, that basically defines the angular divergence of your beam, and most importantly, it determines the maximum size that you can probe. And finally, a detector to detect your radiation, and that basically the use of large detectors is usually better. And in some cases, we use 2 detectors basically for a wider Q range. As I said, the fundamental physics of X-rays and neutron scattering don't really differ, but X-rays and neutrons, their properties differ quite greatly. And as Graham explained at the beginning, so both of them have no charge, but while X-rays have no mass, neutrons have a mass and they have a spin, and that makes them quite unique because that allows us to look at the magnetic properties of materials. X-rays interact with electrons, which makes basically the interaction varies with the atomic numbers, with the atomic number, while neutrons interact with the atomic nucleus, and that makes basically the interaction is a property of the nucleus. And basically, as you can see here, and you saw this before, the scattering length density varies quite randomly with the atomic number. Obviously, they are used quite complementarily, but the choice between using X-rays or neutrons is usually dictated by the problem and by the availability also, but mostly by the problem. So while X-rays are readily available, mostly as lab sources, neutrons are not. X-rays have massive fluxes compared to neutrons, and that is really very useful to study time-resolved systems, especially in kinetics, but also in systems where we have very little material, such for example, in biology, that's widely encountered. Neutrons are only used in large-scale facilities, but the biggest advantage of neutrons is basically their sensitivity to light elements, but also the fact that they are very highly penetrating and that they allow for the measurements of magnetic properties. Also, a big advantage of neutrons compared to X-rays is the lack of radiation damage, and also the use of contrast variation. And that's something I want to tell you a bit more about. So, as I said, there are really many experiments that really do not require the use of neutrons, but not when you have a multi-component system, because there you will use this big advantage, as I said, of contrast variation. And contrast variation, because of the difference in scattering length density between hydrogen and deuterium, what we can do is basically by varying the amount of D2O and H2O in the background or in the solvent of a sample, we can match the scattering of some of the one component to the background, and hence we accentuate basically the signal from one component, and that way we can look at the scattering from the different component, but also at the scattering from the cross term, and that's widely used especially in biological systems, such as, let's say, we have a DNA protein complex. If you have your complex in, say, 40% of D2O, you will match the signal from the protein. You can only see the signal from the DNA. And if you have another contrast point at, let's say, 68%, you're gonna match the signal from the DNA, and then you look only at the signal from the protein. And if you measure at, say, two other contrasts, let's say pure D2O and pure H2O, then you will look at your complex in its entirety. And then by deconvoluting the signal, basically the different scattering profiles, you end up with the contribution of the protein, the contribution of the DNA, and the cross term, or the complex, basically. So that's really the biggest strength, at least, of neutrons compared to X-rays in small angle scattering. So what type of information are we looking at from SANS and SACS? So we look basically at the nanostructure of systems. So we look at the size, at the shape. We look at the internal features, and that's basically, we can do that by using contrast variation. We look at the molecular weights and the aggregation number. We look at the surface volume ratio using the broad model. We looked at the interactions in case of concentrated system, and that's the kind of system we encounter quite often in industry. These interactions can be hard, soft, charged. We look at the location of components, again using contrast variation and interfaces. And we look at the microstructure in case of, say, porous solids. So now I'll move to the second part of the talk. So SANS at ISIS. So here is the SANS group. We are 8.5 scientists. So it's the group is headed by Sarah Rogers. There is quite a variety of expertise in the group. So I'm more of a biology person. That's why I talked about DNA and protein. Sorry about that. But basically, we usually deal with different types of sample, especially when we receive express samples. Proof is I characterized the JM sample that Hamish gave me. So me and my colleague James Douch, we look at mostly biological samples. Steve King is rather polymers. Diego and Dirk are rather magnetism. And Leij is rather soft matter. Richard Heenan is semi-tired. He's mostly working on the Loki instrument, on building the Loki instrument for ESS. We have 4 beamlines at ISIS. So we have LoQ, SANS-TD, LAMO, and Zoom. And as I said, Richard Heenan is building the Loki instrument for ESS. It's sort of we have 4.5 instruments. On the left-hand side, you can see the Q resolution, basically, of all the Q range is covered by the different instruments. And as you can see, SANS-TD, because it has the detectors, it has movable detectors, so it can cover quite a wider range of Q compared to the other instruments. But basically, using one or the other of the instruments is dictated by the problem at hand. So on Zoom and LAMO, you can do polarized SANS, while you can't do that on SANS-TD and LoQ. On LAMO, you also can do what we call SANS. That's spin echo SANS, that allows you to look at larger structures between basically 30 nanometer and 20 microns. And that brings me to the sizes that are traditionally studied on SANS instruments. Traditionally, we study sizes that are between 1 and 100 nanometers. Our resolution doesn't go below 1 nanometer. But SANS allows us, SANS allows us to go to higher, to look at larger sizes. And as I said, it's between 30 nanometer and 20 microns. But it's quite a relatively new technique. There are only 2 instruments in the world, so LAMO at ISIS and another instrument at Delft University in the Netherlands. Okay, so obviously there is a variety of sample environments that can be used with the instruments, because what our users want is mostly to look at their samples at conditions that are, let's say, in situ, as comparable as possible to the real conditions. So we have different types of cryostats, furnaces, and magnets. A sample changer is our most used sample environment, and with temperature control. We have Lincoln stages for advanced temperature control, and bryometer and shear cells, pressure cell, T-jump cell for studying non-equilibrium phases, in situ DLS and UV-VIS. We also do a grazing incident SANS. We do stopped flow to study mixing kinetics. And we also have well-equipped offline labs that allows for further characterization of your samples using whether X-ray sets, AFM, Brewster angle microscopy, spectrometers, multi-angle DLS, and a range of other techniques. So in this slide, it's a overly charged slide. I put examples of the science that we do on the SANS instruments with the sample environment that it uses, and you can see that basically it's a variety of science that is explored, that goes from biological systems to soft matter, to hard matter, and to industrial systems. A typical month on SANS TD, I will usually deal with chemists looking at colloidal crystals to nanoparticles in my cells and microemulsions. I'll often have industrial partners coming to look at chemicals such as perfumes in my cells, or in situ rheology of pharmaceutical formulations. I often deal with biologists looking at solution structure, or say growth of fibrils in diseases, with pharmacists looking at the movement of drugs into vesicle bilayers, with polymer scientists, with physicists looking at structural and magnetic scattering from superspin glasses. So it's quite a huge variety of science that's done all the time. However, the landscape, or the scientific landscape, is changing a lot. That's what we noticed during the last few years. And we are rather moving towards large length scale compared to the 1 to 100 nanometer that we were used to during the last 10 to 15 years. So we're seeing more and more industrial formulations that are quite concentrated, but also that have larger scales and different kinetics to it. And hence we need to change our, we need to evolve with this evolving demand for large length scale systems. So, and finally, I will tell you a little bit, I will give you 2 examples of contrast variation and contrast matching use, where in one case it worked and it led to a PNAS paper. In one case, we're still struggling to try to understand the data, and Hamish knows a bit about this. So a typical experiment on a SANS instrument will give you a 2D pattern that basically you will radially average, you will have a scattering profile, a 1D profile, that you can't do a lot with, apart from doing some basic analysis that gives you the size maybe, but that's about it. To have a more like proper quantification, you need to do modeling of your data. And to model the data, basically you have to quantify the information that you have in your signal, and your scattering is basically has information about the form factor of your particle, and that's basically the intraparticle information. So it has information about the size and the shape of the particle, but it also has information about the interparticle positions in the system, and that's what we call the structure factor. So modeling the data is usually very dependent on the problem at hand and on the system at hand, and hence the softwares that we use for modeling the data depend on the problem that we have. For some systems, we can use geometrical models in case we have extra information that is usually provided by microscopy or by dynamic light scattering or other characterization technique. In case we don't have that, we usually use empirical models, and you will see an example in the next slide. So as I said, the most important advantage of SANS over SACs and SALS is the fact that we can use contrast variation, and we use contrast variation to look at lipid nanoparticles that are used to for mRNA therapy, for cancer treatment. This is actually an industrial collaboration with a pharmaceutical company, And what happened here, we used contrast variation to look at the structure and the location of the different lipids and the mRNA in this nanoparticles. And we can we were able to characterize these nanoparticles basically using a core multi-shell model, and this is a geometrical model that is available in a software called SAS View that is supported by ISIS and several other facilities around the world. This was a successful use of contrast variation. A second example is here, and this is a very recent data from a sample, a Johnson-Matthey sample that Hamish gave me. And we tried to do contrast matching because this was an express measurement. So usually in express measurements, we are limited to the number of samples that we can run. And what we tried to do is basically to match the contrast of the core particle or the scaffold particle. But unfortunately, we didn't seem to have succeeded at that. As you can see here, there is barely any difference between the two signal, the one coming from the pure powder and the blue one is from the powder soaked in or wetted with a mixture of 60% H2O and 40% D2O, which was supposed to be very close to the match point of the scaffold particle. So so basically, yeah, we couldn't get any information out of this contrast matching experiment. So this is basically, this illustrates the difficulty of running a contrast matching experiment. Usually contrast variation is a much easier way of getting information out of your multi-component system. So that brings me to the end of my talk. So basically, small angle scattering probes length scales from 1 nanometer to 100 nanometer. But as I said, the landscape, the scientific landscape is changing, and we're moving towards larger structures. SAS does not see atoms, but larger interesting features over many length scales in your system. It is similar, it is actually diffraction, but does not require crystals, and that makes it much easier or quicker to run experiments. Applications are only limited by imagination, and SAS can be used alone but often complementary to other methods such as microscopy and NMR in cases of proteins and nucleic acid. SAS data analysis is very application dependent, using a very diverse set of approximations, models, and software. And that was about it. So thank you for listening. And if you have any questions, I will be happy to answer them.